Your, I advertised your talk widely at Stellenbosch, and I see that none of them have logged in yet.
I see some of my colleagues are logged in. I
Are they?
think so, yes.
Okay, good. Good. I'm not recognizing them. Who is your colleague that's logged in?
I think Catherine has logged in. Catherine
Oh,
S.
I see her. Yes, I can see.
Hmm.
But Ben Lewis didn't come in, and none of the biochemistry lot. What a
So Trevor, whenever you're ready to start, you can go ahead and I'll start
okay.
the recording.
Okay, so what's the time now? It's exactly on time. Well, that's very prompt of us.
Thank you.
Okay. Okay, you're starting the recording.
I can right now.
Good afternoon, everybody. It gives me great pleasure this afternoon to introduce Christian Muller-Niederbach. He's a professor in the Department of Physics at Stellenbosch University in South Africa. His main specialization is the theory of soft condensed matter physics and the physics in biological systems, approached from both equilibrium and non-equilibrium perspectives. His work models the physical properties of networks of filaments, the role of polymer entanglements, and the role of active transport processes, such as molecular motors. He's taught a broad range of physics at both undergraduate and graduate level. I should also comment that he's the representative on C6 Biological Physics Committee of the International Union of Pure and Applied Physics. So he's our representative in the world. So thank you very much for agreeing to talk this afternoon, Christian. And I think I'm just going to say your title, The Physics Associated with the Cytoskeleton, Networks, Motors, and Confinement. And then it's over to you. Thanks, Christian.
Thank you very much for the introduction, Trevor. Thank you very much for the invitation, and I'm delighted. Thank you also for all of you who are joining this presentation. I hope that you can hear me well.
We can hear you.
Great. Um, so indeed, uh, I apologize to those of you who may have heard large sections of this talk before, but I'll be speaking about a large sort of cross-section of work that's happened over a number of years, with whom a lot of people have been involved too. Um, on the slide that you see in front of you, you will see that there's a large number of former and current students and collaborators: Somiaro Azote, Stana Pashong, Nadine Dutoy, Cornel de Villiers, Christo Roos, Arash Azari, and colleagues such as Ben Loos, Andre Dutoy, Jürgen Kriel, and Giuseppe Pedicane, who've been involved with various bits of this project. And I'll probably also be citing papers with people with others during the course of my presentation. As a physicist, of course, I'm interested in the real physics properties and perhaps how they can help with understanding aspects of biological systems. And here are some interesting images. I didn't make any of them myself, except now. Um, Unless if I can get the laser pointer going. Yeah. Um, uh, Sorry. Things look a bit different than I tried earlier. Laser pointer, there we go. Except for this image where I was involved in this, these are images, the ones in red that you see in front of you are made by Ben Loos and Andre de Toy, the Department of Physiological Sciences at Stellenbosch University. These colleagues were looking at putting cells on substrates where they confined spatially these cells that grew. And what you see in red is the tubulin cytoskeleton. And one of the questions that I'll be addressing in this talk is, as in the second slide, the second image over here, is that they managed to change these substrates to confine otherwise identical cells to ever decreasing radii, going from 50 micrometers over 40 micrometers to 30 micrometers. And already on this slide, you can see what I'll generally be talking about in the first section of my presentation is that as you take a look at the image of the red stuff, the microtubules, as they reduce just simply the diameter of the cell as it lies on their microscope slide, you will see that the location and where these microtubules, where the microtubules bundle changes with the amount of confinement or the radius of the cell. So this is the kind of question that one wants to address, and I was delighted that they performed this experiment. So the other images that you see were parts of our experiments to confine growing microtubules in different contexts done at the Appleton Rutherford Laboratory in near Oxford in the UK. More importantly, but another context also from the work of the group of Ben Loos at Stellenbosch University, done by Jürgen Krill, was also to think about these cytoskeletal networks. So over here, you would typically see, again, this kind of red image of one of these confined networks, but you will also see other parts of this image where they have lysosomes and they have autophagosomes that are organelles associated with recycling certain materials in the cells that are moving probably or directed by these networks in the cell, where we want to take a look at how are these networks associated with the motion and the and where these organelles, such as these blue and the green autophagosomes are. And one can certainly measure currents and look at the structures of these kinds of networks. So once again, the physics of the cytoskeleton is also, really how it leads to the arrangements, perhaps by various kinds of mechanisms from a physics perspective, of particles physically in different parts of the cell. This in physics or soft condensed matter physics terminology is really thinking about microtubules or many of the other filaments that are around in the cells as forming some kind of networked structures. They don't necessarily always form networks, but we generally think about some kind of network structures. And this has been a very long study in polymer physics and soft condensed matter physics, usually associated with thinking about the mechanical properties and of such systems. So over here again are just a few cartoon images and one famous sort of Wikipedia image from microscopy of how one can think about different types of networks. So at the top at the top, you see a kind of cartoon of something that we would call cross-linking, and you'll hear more about that later on, is where you essentially use some kind of process to glue or stick two different filament or polymer-like objects together. It's one type of networking. And down below, and we'll also discuss this a little bit, you see another kind of networking where you see a kind of tree-like branched structure that grows. And these networks might have completely different mechanical and other properties, how one thinks about them. So I've already alluded to these objects that are called microtubules. Here, once again, is my cartoon version of how they look. They are assembled in a helical form of dimers to an object that has a diameter of about 24 nanometers. And on the length scale of the the typical cell is a relatively rigid object. These are quantities measured by something called the persistence length. We will also be dealing with actin filaments that are much more flexible and a little bit thinner, but also assembled and form some kind of helical structure in the cell. What is going to be even more important towards the end of my talk is the fact that, if you compare these two diagrams, this one where I've just sort of assembled these spherical particles in my cartoon, over here, the spherical particles have been replaced by cones. And this replacement is to somehow illustrate to the viewer that these filaments have a sense of direction. So there's one end that is completely different to another, or for example, when they're used in possibly transportation, that this might indicate a preferential direction of travel of machinery along them. But let's go back to that first question that that I showed, where I showed you the slides where the cells were confined to different sized regions. And what I'm be showing you here is a 10-year-old computer simulation done by a postdoctoral research associate, Arash Azari, where he looked at confining certain polymeric particles. They're not necessarily biological filaments, but polymeric abstract particles in a computer simulation inside a spherical region. So the spherical region is shown here in terms of this gray region. The particles themselves consist of relatively rigid sections joined by flexible bits, and again a rigid section or relatively rigid section joined by a flexible section. And the only difference between the different colored chains that you see in here, so there are these 4 different colors from the light blue to the dark blue via the red and the sort of brownish yellow, represent chains that consists of identical monomers in all of the cases, except that the length of these rigid bits or slightly more rigid bits than the flexible bits differ. So, for example, the blue chain has the longest rigid bits before it bends around, as you might see here, and the light blue one is the most has the most flexible bits interleaving very short rigid bits, and the red, etc., in between. And I simply want to show you an animation, which was sort of various snapshots of the molecular dynamics simulation of how these chains move around in a confined region. And as we wait for the animation to evolve, you will see that the size of the sphere shrinks. And somewhat later in the simulation, there's another shrinkage that happens, and you will see a snapshot of that, or part of that happening now. And finally, in the most confined region, you will notice that there's quite a strong difference in terms of the conformation or where the polymers go. And you will see that the most rigid segments actually will sit outside on the surface, encapsulating the more flexible chains in the middle. We have to remember that the only differences between all of these different chains of the different colors is that they have different lengths of rigid segments. All of the little spheres, the particles, have exactly the same kind of pairwise forces between any of the 2 pairs of polymers or any pair of polymers. So the only difference really is in terms of that stiffness of these segments. So this can, of course, be expressed in terms of other graphs. So what you see over here, for example, is an illustration of the density as a function of the radius from the center of the sphere under conditions where you're making the radius smaller. So circled at the end of all of these graphs is just simply that final radius of the sphere as we go and confine the system. So the relatively unconfined system and the color scheme follows the color scheme of the chains that you just saw. So the bluest chains have the longest stiff segments, and the light blue ones are the most flexible ones. You see most of it sits in the middle, and then there's very little towards the sides. But as you make the sphere smaller, so here the sphere just has a radius of 14 units, you will see that all of the blue stuff sits mainly on the surface. So there's a real change in where these things go and sit as you make this system smaller. So that's one of the ways that we want to look at it. And now we want to think about how does one address this, or what would one of the possible ways be of addressing this in terms of theoretical physics. And one way to think about this is to think about filaments that assemble or self-assemble or grow or have a variable length, as you will see over here, cones just joining this kind of filament, and establishing a kind of theory that allows us to deal with the growth of a filament inside a confined region, as you will see. The gray zone represents the forbidden zone, where there may not be any filaments, but where the filament has a certain element of stiffness. So that means that you have one bond pointing in a certain direction, and it costs a certain amount of energy to point in a slightly different direction. So it costs more energy the less straight that bond is. So the theoretical tools are really quite simple, and they're nice to write for a theorist, because one can use sort of almost quantum mechanical kind of language for this, but it's still a pure statistical physical kind of question, thinking about the statistics and the configurations that such a chain can have. And the summary of this slide is not really that you're supposed to be able to read all of the equations, is that we can establish a set of coupled integral equations represented here in a diagrammatic way that allow us to figure out, how filaments are oriented and where most of the filament goes to sit inside a confined region if one were to solve this. So in very special cases, one can solve this by hand or analytically and write down an equation. In most cases that are interesting, one will have to turn to a kind of numerical approach for this. And so what it helps us to do is to figure out these functions that are represented by the Greek letters psi, in the end, that tell us something about the density. Where is how much of a filament in a confined region that we have? And this is what several students have been working on, also in related features. But here's just a brief summary of what happens. And again, I'd like to just recall to your memory the images that you saw of the confined red tubulin inside Ben Laws's cells that were confined to smaller and smaller radii. So there are these measures that you find. That here is, for example, density plot for these chains confined to such a region, and you will see that under certain circumstances that I'll explain in a moment, you will have most of the chains sitting relatively smoothly towards the sides of the system, and sometimes even more extremely so. Sometimes if the chains are short or if the chains are bendy, you don't get a situation where most of the chain sits towards the perimeter, but actually in the middle of the system. So these are just simply the extreme kind of cases that you can think about confining these kinds of chains, and the result of the kind of calculations that you have. So in the first case, in the first instance, this mimics what we see in the rather more complicated experiment that Ben and his students looked at. So let's just try to understand, what is happening over here. If one has relatively short chains, such as in this cartoon over here, consisting just out of 5 bonds, and they're rattling around in some kind container here drawn as a box, but it doesn't necessarily have to be. And you think, on average, where do these short chains go and sit? They actually have a relatively flat distribution. Okay, so they can statistically go and sit anywhere, and they slightly avoid the walls a little bit. And so that would be the kind of result of a calculation that my student Somialo Azote, a former PhD student Somialo Azote, found. She would also find that if you have an extremely flexible chain, again illustrated by this rather poor cartoon over here, some very bendy object, and you confine it in the box, it still looks as if it's very homogeneously distributed. But if you have a very stiff chain, okay, this very stiff chain, and you put it inside the box, it curls around like trying to put a piece of wire into a confined kind of region. And this, again, is just borne out by using that formalism with the complicated looking equations that one has. So inside a box, you can figure out that it goes and touches the walls with a very high likelihood at the edges, or in a circular or spherical region, the density is increased towards the sides, again mimicking the kind of diagrams that Ben Laws's lab produced, or the images that Ben Laws's lab produced. So to a certain extent, this really works. One can then, of course, start thinking about more intricate geometries and start exploring this numerically in a variety of different ways. And this is what Chris Duchos did in his MSc thesis a couple of years ago, taking a look at the distribution of these chains inside, two-dimensional regions of a certain type, and he looked at a large variety of them. The slide is very busy. Again, it just simply shows distributions of where chains goes, where he has numerically been solving these kinds of equations. But I'd like to point out important measures that we typically use in these chains. I've referred to the stiffness of the chain, and the stiffness of the chain is typically expressed through something that's called a persistence length, which is the length scale over which there is a correlation between the orientation of bonds in this kind of system. And the criterion, or the other length scale in any confined system, so here's some of the spheric or circularly confined system, is actually the diameter. So we're generally comparing the length scale of the persistence length of these filaments to the diameters. And so this can be done for boxes or certain dimensions of these systems. And what Chris did was actually take a look at where shorter chains, where the typical length is 25 steps, versus longer chains that towards the bottom of your image go and sit. First of all, in a density distribution, but in another sense, what he can also figure out is essentially the orientations of the chain. So do the filaments align parallel to the sides of the container, or are they perpendicular to the sides of the container? And whereas this can be done in 2 dimensions in a variety of different scenarios, the kind of thing that really interests us is what happens is if you don't want to just put individual chains inside there, where I think we know how this works very easily, but what happens if you want to put tree-like structures inside this kind of confinement? And in this case, again, we're dealing with rather theoretical trees in our formalism, but these are inspired by thinking about actin networks that can form in the cell. So what you're seeing here, again, is an animation of a set of actin filaments where the actin filament grows, but where a branching process can occur through the so-called ARP2-3 complex of proteins that can branch at an angle of around 70 degrees off the side of any filament. So you would have seen this filament growing, and you would have seen that one being seeded off here at another angle, growing off in another direction. And so what you find is this kind of tree-like structure that grows, but without cycles in the system. And again, I just want to contrast it over here. This is different to the kind of idea of where we use the terminology cross-linking, that is essentially sticking together or chemically bonding to different filaments by usually a third particle, but they can also link directly. So we are trying to explore these tree-like networks and think about how would one of these how would one of these sort of slightly physics abstract kind of tree-like structures actually, when branching inside a confined region, look, and what kind of forces might one measure if one were to deform that branched region? So once again, you will see here sort of diagrammatics that represent the maths of the formalism that one can solve in order to understand this. And in this case, rather than thinking about a formalism as above here, where one can construct all possible sets of linear chains inside a confined region by a kind of hierarchical equation by using this and substituting that into this, and you get a sum of all chains of all lengths and all different shapes. For branching, you would then start thinking about the possibility of a filament growing, but also including side branches. And if one were to write this out, one does the typical thing that one would do in statistical physics, is sum over all possible kinds of trees that can emerge, and of course all possible shapes of these trees. So for example, this recipe will produce all linear filaments, but all ones that are branched after the first step, and all ones that branch after a second and the first step, etc., etc., etc. And then of course we solve these equations where it's in some or other confined regions. So we're looking at how can one start thinking about the trees, that tree-like structures that grow in a thermal environment that have some kind of persistence length and stiffness of the branching. How do they look if they grow in this kind of confined region? And so this is what Somialo Azote took a look at in great depth in her PhD thesis. And then there are these interesting scenarios to take a look at. So there are different things that can happen. If you think about, a tree that grows inside a confined region, either it's some kind of floppy tree that just simply curves around and fills the whole region, or if it's a very stiff tree, it might stop growing because its branches grow perpendicularly into the sides of the walls. So, over here, so for example, you see one of these systems, which was worked on which Somialo worked numerically, where the tree has grown. Most of it is near the sides of the container, but also the tree is so stiff that its branches actually stick into the walls. Whereas if you take a look at other situations, you might find over here that the tree actually curves around and forms a significant fraction of branches that actually bend around to be parallel to the walls. This one can explore with the parameters that are associated with the bending and the bending of the filaments and the strength of the branching itself. And so depending on whether one is in 2 dimensions or in 3 dimensions, one can identify different regions that depend on how likely it is that the filaments grow versus how likely it is on the vertical axis there is branching. And this can be discussed in terms of how the curvature of this density profile looks. And so we have these 3 regions. They're not quite as distinct in 3 dimensions as in 2 dimensions, but the green region means that the chains are mostly near the walls, but perpendicular to a radial vector for a spherical confinement, whereas if you change the parameters a little bit, chains are mostly near the walls, but anti-parallel, so they're curving around like that bit of wire that we originally saw. And here we just have sort of, in the bottom region, we just have sort of homogeneous confinement with a slight density maximum in the middle. So we can start thinking about where these networks go. But and we can also put them, as Christo did in his MSc thesis, start putting them into really complicated kind of geometries and figure out where and how they align. So those images on the right, slightly more sort of complicated ways of figuring out where most of the filaments go and whether they're aligned in a certain geometry. So here you see the images for this final sort of dumbbell geometry, the C geometry that you have. But all of this is now sort of relatively straightforwardly numerically accessible in terms of this. But what happens if I put one of these tree-like networks inside a box and I start, simply shearing this box or deforming this box? What one can do is one can deform it very slowly, and one can figure out what the cost is to the free energy of the network. In a certain sense, one is then asking, how much energy do I need to deform this? And you're actually trying to figure out a modulus for the network of deforming this, shearing the box or changing the shape of the box. And so doing this by changing the shape of the box, one can figure out in terms of the dimensions of the box, changing the dimensions of the box, you can figure out what the modulus is of this, and you can see that the modulus increases with the amount of with the amount of polymer chains in this system. We can also shear this box. So actually over here you have a box, and you can see by the diagram that it's some kind of rhombus over here, that the region's shape has been changed. So I'm slowly, collapsing some box in which these networks grow. The networks in this instance are concentrated towards the surface. And what you then find is you can actually also calculate the modulus of these systems as you go and shear this gamma. And you will see that shearing it to a certain degree increases the shear modulus, and then it decreases again. So here are just simply rectangular and square cells, but you find this curious peak. And the interesting interpretation that came out of this work was that this can certainly be interpreted as the point where a kind of strut is formed across the diagonal of this box. So all of this can be associated with real sort of mechanical properties of this. It's further possible to think about what happens if you have more complicated and further sort of polymer physicsy kind of interactions between all of these filaments. How do they change? What happens if you combine stiff and less stiff chains in a certain region, as you would have seen in that computer simulation right towards the start. But I'm running out of time, so I'll just skip to the next bit. So we've just seen that one can deal with tree-like structures in a confined region and learn something about the mechanical properties and how the bonds are oriented and distributed within this region. A completely different and probably even older kind of polymer physics concept is the idea of cross-linking, where we glue two chains together. So I don't know whether I've got an animation which works over here. No, if you glue these chains together, either by a cross-linker of some type or just by chemically bonding them directly, as you would see over here, these structures start having completely different mechanical properties than one might have seen for the tree-like network. Because, well, of course, these blue plates represent the rest of a complicated network of chains. But what one would then typically find is that, of course, if these chains start fluctuating and moving, their motion and their fluctuations are coupled via this crosslink to at least another 3 segments in a complicated way where they're all affected by this. So in statistical physics, the kind of question that one needs to answer in order to describe some of these networks is that one also needs to figure out, in how many ways can such networks form, and these cross-linkers then form constraints. So if I just show this animation again, you just see that there are two different ways in which these two cross-linkers can connect to a chain. They would both perhaps happen in a physical system. But once these cross-links are formed, like when these cups meet these spherical counterparts, they form a constraint, and this constraint on the network is a different constraint than that constraint on the network. So the mathematical task for describing a polymer network is not only describing in how many ways does the network need to be able to form, which is the basic statistical physics question, but through forming it, one is also enforcing a constraint in that now the green chain is linked to the top part of the red chain in the bottom image, whereas the green chain is linked to the bottom part of the red chain in the upper image. So there are a variety of ways to do this, but one way is to enforce all of these possible ways of pairing, and mathematically one can try to do this by using Dirac delta functions in the formalism. And there's a really beautiful field theoretical approach developed by Sam Edwards, many years ago, so you see 1988, but also in a similar method by others, that uses the mathematics of Gaussian integration in order to do that. Now, I'm not going to go through all of this Gaussian integration, but it happens that if one uses one of these sort of very standard kind of functional integrals, one can, by evaluating this complicated integral or this integral that looks strange on the right-hand side, which has these fields phi and phi star in it and the various sets of coordinates, actually create a mathematical function that pairs each lowercase r that sits within a phi to each uppercase r that sits in the field phi star on the right-hand side in all possible ways, so all possible combinations, and enforcing these constraints. So this mathematical function simply ensures that it enforces the constraints of the cross-linking, but it also ensures that we are actually summing over all possible ways in which one can do it, which is responsible for a statistical physical sum. So in this sense, we can almost start thinking about the role in this diagram over here of the fields phi star and phi as different particles. So the fields phi star can be attached to the cups, and the fields phi can be attached to these little balls over here. And therefore, you can match the cross-linkers, in this case, with various functionalities in all possible ways. You can match the cups to all of the possible attachment points to the polymer chains. So that's mathematically doable. It leads to some really surprisingly non-linear field theoretical expressions. But as Nadine Dutoy, who completed her PhD just a couple of weeks ago, showed, is that indeed, although this is a non-linear kind of theoretical construct, one can really systematically go through it, that there's always a solution to this field theory that is physical, and always one solution that is exactly physical, no matter how non-linear and weird these field theories look. And that gives a very sensible way of thinking about how to enable this kind of cross-linking in the mathematical formalism. From this, Nadine, for example, is then able to predict various kinds of measurable things about these kinds of polymer networks, for example, such as structure factors. What Nadine's theory also does is that these ideas of using these Gaussian integrals is extended to thinking about these systems as dynamical systems. So we can really start thinking not only as on the statistical physics or equilibrium statistical physics of polymer chains, but we can actually adapt this to chains that diffuse in space, are modeled through Langevin equations, where you have crosslinkers that are also modeled through Langevin equations, where the field theory then actually enforces the kind of constraints that you need in these kinds of tools to predict this. Nadine, in order to check what turns out to be rather technical calculations, also provided um, uh, computer simulations, molecular dynamics simulations, on these kinds of networking systems, where in red and blue, you have a variety of different chains in a periodic box, as you would see. And in yellow, although quite difficult to spot on this image, are possible crosslinkers, particles that can move around, that can sometimes crosslink the chains, but then also stop crosslinking the chains and diffuse around in space again. So really dynamical mobile crosslinkers that temporarily cause a crosslink between blue and red and blue and blue and red chains. What you see on the left is the result of a simulation. There's, I think, 300 chains, but I speak under correction, where this cross-linking has been switched on. And what you will immediately see is a kind of structurally, there are these gaps and holes as the cross-linking makes parts of the system collapse. Whereas if you have exactly the same particles, but the cross-linkers are sort of switched off in their cross-linking action, you have a seemingly far more homogeneous looking kind of snapshot of this kind of system. And indeed, this can, of course, be related to thinking about what happens if one does some kind of light scattering from these systems, and or any kind of scattering from these systems. And one immediately sees that there's a difference between, here's a cross-section of the dynamic structure factors, in these cases, at a fixed frequency, for Q, the momentum transfer, magnitude of the momentum transfer, and you will see that the same system, when it's cross-linked versus when there's no linking, really differs in terms of the structure factor and the features that we see. So indeed, even complicated theories and these networking approaches for dynamical systems work, and we really wanted to develop this kind of tool to eventually describe what happens in biological systems where there is dynamical version of cross-linking for semi-flexible chains. I need to mention that these simulations also include the semi-flexibility of these chains. So one can deal with this kind of cross-linking phenomenon in computer simulations, but also in an analytical theory. This kind of approach used many years ago also to think about clustering of particles. Again, you see the usual sort of tools of little cups that can cross-link to other cups. And here's an example of how an equilibrium field theory of this looks. And for any of you who might have been ever dealt with these kinds of theories, it really does look scary. There's a field phi and a field phi star, and then there's a field phi star inside a logarithm, and then some really high powers of these fields. But it turns out that even though it looks so terrible, at least in some approximation, this seems a reasonable approach to take. One can also use this in a combination with the confinement theories that I described beforehand, where certain updates to just simply the fugacities and the kind of linking of these systems is possible. However, I want to introduce the idea of molecular machines to you. Again, as in a biophysics colloquium, active systems are probably very familiar to you. And again, I'll be looking at them from a rather naive perspective of a theoretical physicist. So here is my very cheap cartoon of a kinesin walking on a microtubule, taking a step forward, progressing along a microtubule. And I'm pointing out again that we have to remember that these filaments with respect to these motors definitely have a sense of direction, so that there's a sense of direction of stepping of this kinesin. The block, I suppose, represents in some abstract sense one or other cargo that can be pulled on by the kinesin, and so it can step forward. Another cartoon is of different motors, a myosin on an activant filament. And here you'll again see those cones that I showed you previously, just to indicate that these filaments have a sense of direction. And the myosin motors, such as the kinesin motors, of course, use chemical energy to drive to change the conformation of the motor, which then attaches and reattaches as a non-processive motor and can actually exert motion or force between the motor head and the filament itself. Again, this is definitely not how it looks in reality, but just simply my physicist's animation of what might of how one could think, at least in a very simple-minded modeling perspective, of the mechanism of such a myosin motor that is pulling along such a filament. How does one deal with this in theory, and how does one understand various effects of this? So a while ago, we were trying to look at the idea of so-called motility assays. With a colleague, Shiladitya Banerjee, and a colleague, Christina Marchetti, many years ago, where we're thinking various of these motors that are linked on some or other substrate. So actually, you have to imagine this as a two-dimensional substrate. My cartoon only has a one-dimensional substrate. We have our filament, again, depicted by the cones on top of it. And these motors that can attach or detach. And when they attach, they want to process in a preferential direction, in the direction of the cones. And if they do that, there's a certain kind of motion that one can, again, predict. So let's hope I get my animation working. So as you can see, the filament is moving and the motor heads are moving in the opposite direction, pushing the filament backwards. But then if the force gets too large, the motors can detach. Eventually they can reattach and then start their motion. And so one can actually start in this space, start dragging a filament through space as it is attached to these motors in the so-called motility assay. I urge you to go, for example, and look on YouTube. There are many interesting movies of groups throughout the world who actually have made very interesting experiments with motility assays and the collective motions of these filaments. How does such a motor motion affect the idea of a network, though, is the question to which I want to return again, since network is one of the questions that I'm focusing on in this presentation. And imagine now that we have two filaments. They are again directed filaments of some other type, but now we have a so-called active crosslink connecting these two. So there are motors over here between this end of the crosslinker and the filament, and there are motors over here. And if we switch on the animation, so these motors want to move the crosslinker in that direction, these motors want to move the crosslinker in that direction. And what we then get, sorry, excuse me, is that we get a real strange coupling of the kind of fluctuations of these filaments and a completely different transmission of forces and perhaps a completely different mechanical response than in the kind of passive networks that I explained previously, even in the dynamical ones. So this has been a lot of work in trying to, over the years, in trying to understand the role of having these so-called active crosslinkers that have preferential directions of motion in certain systems in networked systems. And one can actually start thinking about how to calculate new or updates to the elastic responses to these systems due to the motor forces. So again, here are some equations, but they just represent the first terms over here with the lambdas like this, represent the usual elastic responses, whereas terms proportional to f squared tell us that the elastic response is then modified by the motor force of these active motors, these red bits that want to pull in separate directions. This has an interesting, possibly, let me rather say it this way, possibly interesting effect in certain kind of systems. During cell division, contractile rings form of actin filaments, and there is a truly interesting and fascinating experimental paper by Kamasaki and co-workers from 2007 that studies contractile rings. And what one finds is that these actin filaments form a contractile ring. In the contractile ring are definitely motors. There might be some other cross-linking particles. And the contractile ring then pinches off the membrane of the cells during cell division by changing its shape and getting smaller until eventually, the single original mother cell has split into two daughter cells by this physical process, in part, of the contractile ring actually, that forces the separation of the cells. So this entails actin filaments and possibly active cross-linkers. So how does a physicist go about a minimalist kind of thought experiment in this kind of situation? Well, what Kamasaki and co-workers also figured out by extremely clever microscopy was to figure out that there are actin filaments. And remember, once again, these actin filaments have certain arrows associated with them. So blue and red representing different directions. So in this kind of ring, you have filaments that are either clockwise or anti-clockwise. We just indicate them here as parallel or anti-parallel. Or sometimes there are filaments that are parallel, but they can be cross-linked by these active cross-linkers indicated by the green little cylinder that my cartoon shows. If you switch on the activity in this green cylinder and, for example, its connection wants to move in the direction of the arrow, you have something like this. And the bottom connector of this cross-link will want to move in the direction towards the left on the red filament. And so the filaments themselves will shift eventually, and therefore they will be moved past each other by such a cross-linker. So an active cross-linker for filaments of different orientations wants to push the filaments past each other. Whereas if filaments are in parallel, what happens is the active cross-linker wants to move towards the right in both of these instances. So the active cross-linker just moves along. All it does, it moves along until it probably falls off or gets stuck at the end. And therefore, its only real role in terms of these filaments is to stick them together, but it's not really moving them relative to each other. So thinking about this, thought, what is one of the potential mechanisms that we can have to think about these active cross-links between parallel filaments? So certainly, the more these filaments overlap, the more space there is for active cross-linkers to attach. So if there's a large overlap between two filaments that are almost in parallel and a short distance from each other, there's the possibility of many such motor pairs occurring, and therefore a stronger force. The less they're overlapped, fewer, and of course fewer still if the overlap becomes minimal. So we have some kind of these filaments being pushed past each other, but the force changes depending on the degree of overlap that these filaments have. So here is a very simple kind of computer simulation that now takes a look at what happens if you put a bunch of these filaments together. So red and blue are just different orientations, clockwise versus anti-clockwise. And I've highlighted one of the red ones and one of the blue ones in yellow and green. And if one follows this animation for long enough, there are these forces, as I've just explained in the previous one, between all of these filaments, between the parallel and the anti-parallel ones. And what one sees that is if one follows this movie around for long enough, the blue and the green and the yellow highlighted filaments actually move in opposite directions around the circumference of this ring. So they're being transported in opposite directions with their opposite polarization. So this certainly happens so that there's motions of these filaments, and the motors seem to push them around. But one can also write an analytical theory of this. Um, the question to us was, what happens in this kind of motion that's illustrated in this kind of movie, but how can one ensure that this ring actually has a force that causes it to contract? Now, again, there are various mechanisms that are proposed, but here's our proposition. If we're thinking about this, all that the activity seems to be doing is actually just simply transporting the rings around in opposite directions if they have opposite orientation, the filaments around in opposite directions if they have opposite orientations. However, there's an important clue to what is happening. So here is a very cheap simulation, but I think quite illustrative of this. So on the circle, you have red and blue particles, very difficult to identify, moving around in opposite directions on a ring. And what I do is I have reduced here on a slider the amount of activity, the active force, the force with which the filaments push past each other. And if the activity is low, you will see that all of the filaments therefore concentrate on one part of the circle. That actually means that the integrity of the ring is broken because they're all bundling together. They're essentially just sort of sticking together, and the active force is too little to actually have a completed ring, a structurally complete ring. What you then see also over here is in this animation is that the current, the amount of particles actually flowing around in this ring completely ceases. If I can show this animation again. So if the activity is higher, you will see that there is some kind of sustained current of particles flowing clockwise and particles of the opposite nature flowing anti-clockwise. If I switch off the activity, the ring loses its structural integrity and collapses. There's no longer any current, as you see over here, and the structural integrity of the ring is gone. So in our work, what eventually we concluded, at least from the simplistic model, is that indeed, and this is work by Stanwa Mebwe Bashong, who was a PhD student in my group some years ago, is that if we have this idea of the active of the networking strength and the active force, so blue and red are different amounts of active forces, we can maintain the integrity of the ring by the active force. So that means the active force actually mixes these particles up that allows the ring to shrink in terms of its networking strength. So the structural integrity is, according to our conclusions, at least enforced by the active force. It's not responsible for the contractility of the ring, which is just the normal networking force, but somehow the active force is preventing the system from collapsing over here. So this was just sort of my very fast excursion through a variety of dynamical and non-dynamical kinds of networking in the confined geometry of a ring or in the confined geometry of some spherical or box-like enclosure, and due to work by a large number of very brilliant students and backed up by fantastic work by several brilliant colleagues. And Trevor, I hope I've kept in time, and I hope there are some questions.
Yes, Christian, you're perfectly on time. And I see that the audience has swelled considerably
Oh, fantastic.
since the beginning, and they're from all over Africa. And welcome to everybody that has joined us for this talk this afternoon. I'm going to throw the questions open to the floor straight away, as I don't see any hands up at the moment. Perhaps you can put your hands up and I'll call you in. Has anybody got their hand up? Ah, okay. Lawrence, speak.
Um, so I was wondering about your experimental collaborators.
Yes.
Um, Do you is the ABIC group at UCT and Stellenbosch, are they tuned in to the cytoskeleton dynamics work?
Sorry, I missed.
The the African Bioimaging Consortium? Are they,
Through Ben Laws, I suppose, yes.
okay.
Just for your information, Ben is actually the coordinator of the African Bioimaging Consortium. So he's very prominent and is leading it and is developing the whole network. Okay, so I thought that was a very fascinating talk, and I hadn't realized the implications of simple physics on network formation and the movement of fibers around cells.
Mm.
But in fact, can you just how many of the actual images that you produced are produced and under what circumstances in living cells?
The sort of the living cell kind of images were the ones that I showed you right at the start from Ben Laws's group. The other images that we are producing are either computer simulations or numerical work on really simplified physics kind of systems. There is still, I suppose, well, definitely lots of space to move from the really simple physics perspective to the much more subtle and complicated situation of what is going on in a real cell. Um, I think where your question might be going, or I hope I understand where your question might be going, is that the hope is as we, as physicists, I as a physicist, and we in our group as physicists learn more about the biological systems, what we can do is assist at some stage our colleagues in the biological sciences to perhaps help them differentiate some of their observations. For example, we can try doing a calculation. In this situation, is it a mechanism of type A or a mechanism of type B? We can hopefully predict what might happen under these two circumstances. And then there's the hope that perhaps under ideal experimental and observational circumstances, one might be able to figure out whether it's more likely that a mechanism A or B, or perhaps a third mechanism, is what might be responsible for a certain phenomenon on the cellular level. Trevor, have I misunderstood your question completely?
Yeah, I think that's more or less right. I mean, that's more or less what I had in mind. But I was thinking, if you see something happening in a cell, what you need to be able to do is to to see whether you need additional components to those that you think are playing a role,
Yes.
and to see whether the behavior can be completely modeled on the
Mm.
basis of
Yes.
the kind of parameters that you've talked about.
Yes.
And then see whether perhaps the behavior is dependent on some other protein or
Exactly.
whatever being
Yes.
synthesized
Yes.
at that
Yeah.
particular juncture in the cell
Exactly.
cycle.
Yes. Now,
Okay.
sort of the physics, the physics approach is definitely, if we think that perhaps this absolutely minimal number of ingredients produces the phenomenon, the first question is, does our model produce the phenomenon, and what else can it predict about it? And then I suppose one can hopefully by doing the comparison decide, well, this is not yet adequate. We need to think about another protein playing a role here or another process. So
I mean,
to a certain extent,
one
it's
of
the
the
process of elimination also that plays a role. Sorry.
the one thing would be condensation of chromosomes.
Mm.
So that would be an interesting thing to see
Yes.
how many components you need in
Mm.
order to predict
Yes.
the
Yes.
condensation of
Yes.
chromosomes.
There are definitely physicists and some of my colleagues working on this. I know, for example, the group of Helmut Schiessel in Dresden at the moment have been working on this for many years in terms of the chromosome condensation, etc.
Yeah. Okay. So are there any, I don't see any other hands raised. Okay, well,
I actually did have one question. Somewhere in the mid-presentation, there was an
Yes.
equation of that on one hand looked like a Hooke's law, but had a logarithmic component to it.
Oh, this one
Maybe before then.
before then.
And I was just wondering where the logarithmic term came in, or was that a reduction of the
I
constituent equation?
can't quite remember where it might have been.
There was a natural log.
Oh, there was one over
Yeah, there
here.
we are. Yep.
Okay.
Persistent lift, yes.
Yes, that's for the persistence length. So
Yep.
this comes, so these W zeros and W twos in the way that that equation is written are essentially weights associated, statistical weights associated with bending straight or bending at a certain angle on the lattice on which we simulated this. So the logarithm simply comes because uh, comes from the fact that the persistence length is interpreted as an exponential decorrelation over the length of the filament. So we're associating one bond vector. I don't know whether I can actually write this over here. So the persistence, one of the definitions of the persistence length is the dot product of a bond vector at a certain point along the filament with the bond vector further down on the filament. And we're looking at distances where we're expecting this to fall off exponentially. And so this is just simply related to that exponential fall off. So it's a little bit of a sort of, I suppose, just a sort of mathematical construct. Yeah.
Okay.
when you were talking about the ring formation,
Yes.
I was thinking about bacterial division. And you could kind of make a ring that gets smaller and smaller, and then
Yes.
Finally the two bacterial cells divide.
Yes. Yes.
Is that what you had in mind when you were modeling that as well?
So this is definitely the reproduction of cells where you have a cell membrane, and I'm certainly not a brilliant biologist or would know all of these things, but what happens is once sort of the genetic material has been reproduced and pushed to two different sides of the cell by these esters, as far as I know, I speak under correction. These actin filaments assemble along the equator of the cell, and then this contractile ring starts contracting and pinching of these two cells to form these two daughter cells. But there are other rings and other mechanisms of pinching off that also exist in a variety of different situations. For example, I think mitochondria divide, by again other kinds of mechanisms that also form some kind of snare around what needs to be pinched off. But as far as I think, it's a different sort of set of ideas there.
Okay. Okay, well, any other questions that people want to ask? Okay. Well, with that, thank you very much, Christian. That was a very interesting talk, and I really did enjoy it. And I hope that there'll be other contributions from the other people. I will just say that this is the last talk of this year on this series of talks. We're probably going to resume in February next year. In the meantime, Lawrence, do you want to advertise the conference?
Yes, there is the joint conference of the African Light Source and African Physical Society. Let me put the link in the chat. Hold on, typing. Force.org. And actually, I'm thinking I just had the thought of inviting a speaker who does time resolved Sachs wax, small angle and wide angle scattering. That could be the experimental view of what we just heard.
Okay. Okay, then with that, then thank you very much, and I'm going to close the meeting.
Thank you very much to the audience.
Thank you very much, Christian.
Bye-bye.
Bye-bye.
Bye-bye.